Hey guys, it's Ms. Lenham. We're ready for the next discussion in our lecture, which is over macroevolution. Now, we've been talking about microevolution, which is evolution on a small scale, so we're ready to now embrace the macro part of evolution, which is a larger scale, commonly looking at the formation of new taxonomic groups. This could be new families. This happens a lot if you're talking about flowers. They're constantly putting more flowers into families or discovering new families. It could also be for genera. It could be any of the taxonomic group, but more commonly, this is the one that we're going to look at, is the species. Putting new organisms or creating new species based on differences, etc. So macroevolution is a result of an accumulation of microevolutionary changes or changes in the allele frequency. So we've been talking about microevolution or changes in the allele frequency. So an accumulation of those in populations over time can lead to macroevolution. You're going to see here it's kind of like AKA speciation. Let's talk about speciation for a second. Okay, speciation is kind of what intrigued Darwin when he went to the Galapagos, and he noticed the diversity of all the species. This was something that really Darwin spent a lot of time on and was perplexed by the diversity of species and kind of got into this. So speciation is a process by which one species splits into two or more species. Okay, so it's responsible, like I said, for the tremendous diversity of life that we have today. It explains not only differences between species, and this is kind of interesting, you know, when we look at two different species of organisms, we look at the differences, but more importantly, we look at the unity of life. How similarities between different species shows that they came from or descended from a common ancestor. A perfect example is talking about a flightless cormorant found on the Galapagos Islands is related very much so to flying cormorants that are common in the Americas. And so there's a need to look at those and you know, show differences and similarities in those cormorants. So it's not just the differences, but it's really the similarities. So it's showing the unity and diversity in life by looking at this speciation. Okay, so before we can get into more of this, we have to kind of establish what we mean. Well, what do we actually mean when we say the word species? So believe it or not, there are multiple definitions of a species in different ways that scientists look at species. On this page you see three concepts. These are different ways of looking at the word species and what that means, but all three of these are actually not the primary definition. On the next slide we're going to go through the primary definition of species, but I must share with you the three examples that are you know, looked at by some scientists. So the first species concept is called morphological. And I know that you know that morphological means form. So it's typically used by scientists to distinguish species by looking at body shape, okay, body shape and other features. It can be applied, which is the benefit, is you can use this to look at asexual and sexual organisms, and it can be even useful without looking at gene flow. So this is one way of looking at species, is looking at the morph, or the form of it, and then putting things into groups based on their body shape, based on their feature. You can also, like I said, look at not just sexual, but you can look at prokaryotic organisms like bacteria. Okay, the second example of, or definition of species that scientists use is in terms of its niche. Okay, a niche is like the role in its environment. So it's looking at not just the the role that it has with living things, but also looking at how it interacts with non-living things. It's more of the ecology aspect of it, and it can also accommodate the sexual and asexual organisms as well. Now, I did want to mention one drawback to the morphological model, because you know you always look at like the positives and negatives. There's a drawback to the morphological model, and it's kind of because it's subjective. Researchers disagree on which morphs or which structures, which features distinguish a species, so they can't always agree on looking at those structures and looking at them to distinguish them between each other. So that kind of has a drawback. Okay, the last one that you see listed here, phylogenetic species, and we've been talking about phylogeny already. This one puts things into groups based on sharing a common ancestor, forming that one branch, you know, that tree of life, and it's looking at morphology, but it's also looking at molecular sequences. So it's studying DNA and grouping individuals based on, like I said, their molecular sequences and their morphology. So that's three examples, and I want to kind of just show you this picture over here because it's kind of cool. When you look at the bottom is Microevolution, or these changes in allele frequencies, might lead to changes in color, for example, the body color or the expression of a certain color like that. Whereas macroevolution will be showing you how this thing over time evolved into a different population altogether, or a different species. So we went from one species to another by an accumulation of these allele frequency changes. Okay, so let's go to the next one. Let me see if I can get out of my... Hang on a second. Okay, before I change slides, I wanted to mention with the phylogenetic species concept, the bottom one down here, there's also a drawback to using this definition of a species, because for scientists sometimes it's difficult to determine the degree of differences that are required to indicate a new species, like a separation of species. So sometimes they can't agree on those differences. Okay. Moving on to, this is the primary definition, so put a star by this slide. This is the definition of species that we use today. This is the one that the book uses, so it's called the biological species concept. It was described by Ernest Mayr in 1942. The formal definition is, it's a population or a group of populations whose members have the potential to interbreed and produce viable fertile offspring. I like to use the word, I use this with my pre-AP kids when I say it's organisms that are so similar that they can make babies, that can then make babies, that can then make babies, that can then make babies. So it's, you know, you're in the same species if you're capable of interbreeding. It can be difficult, believe it or not, there's a drawback to this view as well. It can be difficult to apply to certain organisms, number one, because it does not apply to asexual organisms. Prokaryotic organisms, we don't always use this because of their reproductive patterns. They don't always have to be in the same species to reproduce. We've talked about transformation with bacteria before, etc. Plus, there's no way to evaluate the reproductive behaviors of fossils. So if you look at the fossil evidence, you're not considering that with the biological species concept because you're looking at what's current. Okay, now I have a picture of a bunch of individuals on this slide, because I wanted to kind of explain to you that despite the diversity that you see among these organisms on the slide, according to the biological species concept, these are all in the same species because they're capable of interbreeding. So they can all move around the world and meet each other, and if they come into contact, they do have the ability to make fertile offspring. So according to biological species concept, you know, these individuals can reproduce with each other. Now, one thing you have to consider when you're looking at the biological species concept, and it's kind of what holds the gene pool of a species together, which causes members to resemble each other, is gene flow. Gene flow is a big contributing factor to the diversity of species. It occurs between different populations, and it holds the populations together genetically. So absence of gene flow is going to play a really key role in the formation of new species. So for a species to exist and not have a new species form, you're going to constantly have gene flow, or the flow of new alleles into and out of a population. And we already talked about, you know, when you don't have gene flow, how that can be one of the characteristics for Hardy-Weinberg equilibrium. So this is kind of what we're going to focus on in the next couple of slides, is the biological species concept. I did want to mention another key word you could put over here beside this paragraph is reproductive compatibility. We like to say that, you know, members who can interbreed and produce viable offspring have reproductive compatibility with each other. Okay, moving right along. Okay, because biological species, or the biological species concept, because biological species are defined in terms of reproductive compatibility, the formation of a new species kind of hinges on what we call reproductive isolation. Okay, you can see right here the word reproductive isolation. It's the existence of biological factors or barriers that impede members of two species from interbreeding and producing viable offspring. So, like I said, once again, I'll read it again in case you're copying it down. Reproductive isolation is the existence of biological factors or barriers that impede or prevent members of two species from interbreeding and producing viable offspring. Okay, so we're going to divide these reproductive isolation, these barriers, we're going to divide them into two types, prezygotic and postzygotic. Okay, so before the zygote and after the zygote. Okay, let's look at this diagram over here, and we're going to elaborate on these on the next couple of slides. But when we're talking about reproductive isolation, okay, the two types are prezygotic and postzygotic. And so you can see over here in the diagram, here are individuals up here, the pink and the yellow, they're individuals of different species, but they're closely related. So what's preventing them from reproducing, and down here at the bottom showing making viable offspring, it could be a number of things going on. Okay, if it's prezygotic, and like I said, we're going to elaborate on the next couple of slides, so don't worry about copying all this down now. But if it's prezygotic, before the zygote, it could be something called habitat isolation. It might be behavioral isolation. It could be temporal isolation, which are going to prevent the mating from even occurring. Okay, so a couple things that might even prevent these organisms from mating. Okay, if these two are attempting to mate, there could also be two other things that prevent them from making the viable offspring, in other words, preventing the zygote from occurring. This is prezygotic, which could be mechanical or gamete isolation. Okay, now, another scenario over here, this is the second type, is maybe it's a postzygotic barrier, where these two organisms that are of different species, they have the ability to mate, but there's some barriers which prevent them from making viable offspring. It could be something called reduced hybrid viability. It could be reduced hybrid fertility or hybrid breakdown. So there's those isolated barriers before mating. There's some that happen where they try to mate, but they still don't make the zygote. It's not a healthy zygote, or not one at all. And then we've got those that have a zygote, but there's some problems with it. And so the end result, no matter which of those there are, of these prezygotic and postzygotic isolation, reproductive isolation, are down here. We do not get viable fertile offspring. And according to the biological species concept, if we don't have this, then we don't have these things belonging to the same species. Okay, so let's look at in a little bit more detail at the prezygotic barriers. Okay, so look at the title. These are prezygotic barriers. These impede mating or hinder fertilization if mating does occur. Okay, so you can see the line right here. There's a mating attempt here which splits the mechanical from the other ones in the beginning. So I'm just going to take these one by one. If you want to push pause after I go over one or write a few things down on your slides, you can do that. So the first example of prezygotic barrier is habitat isolation. It's when you have two species that occupy different habitats within the same area. They may encounter each other rarely, if at all, but even though they're not isolated by obvious physical barriers such as mountain ranges. So you might have these existing, for instance, in Kingwood, and they're in the same area, but they don't really come into contact with each other. These two that you see in this picture are garter snakes. They're from the same genus, but they're in different species. They live in the same geographic area, but one mainly, the A picture right here, lives mainly in water, and the other one is primarily terrestrial. So even though they're in the same genus, they're both types of garter snakes, they don't encounter each other, and therefore there's not the need to reproduce. So habitat is an isolating factor for these two. Okay, the second one is called temporal isolation. These are species that breed during different times of the day, different seasons, or different years, and therefore their gametes do not mix. In the picture, what you see here is in North America, the geographic ranges of the eastern spotted skunk, which is the top one. Okay, that's the eastern spotted skunk, and then the bottom one is the western spotted skunk. Their geographic ranges do overlap, but one of them mates in the late winter and the other one mates in the late summer, and therefore they are isolated based on when they're reproductively ready to mate. So these two are two different species based on that. Another example of this one too with temporal isolation we see here in North America as well is the meadowlarks. There's an eastern population of meadowlarks and a western population, and they are also temporally isolated from each other. And they leads into the next one, which is behavioral isolation. Actually, I just messed up on that. Those meadowlarks are behaviorally isolated. My bad. Temporal isolation is, there's an orchid species. I got those mixed up. Temporal isolation, there's three types of orchids that live in the rainforest, and they reproduce they flower at different times of the year, and therefore they're isolated from each other based on those reproductive patterns. The behavioral isolation, this one, the meadowlark that I was talking about, is they have a different courtship ritual, and so the eastern meadowlark and the western meadowlark, the female waits on a certain courtship ritual from the male, and if he doesn't display it, then they're not going to happen. It's not going to reproduce. In the picture that you see here, this is one of my favorites. Courtship rituals that attract mates and other behaviors are unique to a certain species, and therefore they're effective reproductive barriers. In the picture, these are called blue-footed boobies, and they're actually on the Galapagos. They mate, like I was talking about the meadowlarks, only after a courtship display that is unique to their species. So if they don't do the unique display, then they're not going to reproduce with them. So these are, and with these guys, these blue-footed boobies, obviously, it calls attention to the female by the bright blue feet. And so that's a behavioral isolating factor. Okay, over here on this side, this is if a mating attempt occurs, there's still an isolating factor that's mechanical. Mating is attempted, but morphological differences prevent the completion of mating. So the shells that, what you see here is the shells of two species of snails that are in the Bradybaena genus. They are spiral in different directions. Okay, I don't know if you can tell on this one, and this one right here, their shell is spiraling in a different direction altogether. And so what that does is it moves inward toward the center. One spirals counterclockwise, one goes clockwise. As a result, their genital openings are not aligned. So they can attempt to mate, like you can see here, but their genital openings don't meet each other because of the shifting in the clockwise versus counterclockwise shell. Therefore, they're not going to mate, and so that would lead these into two different species. Okay, the last example, and I'm putting a little arrow so you can see right here. I forgot this one because it was on the next slide. This is the last example of prezygotic barriers before we get into these postzygotic barriers. This one is gamete isolation. Okay, so once again, there was already a mating attempt. Okay, so you saw on the slide before it, there was a mating attempt. So this one is, example, sperm of one species may not be able to fertilize the eggs of another species. For instance, sperm may not be able to survive in the reproductive tract of females of other species, or biochemical mechanisms may prevent the sperm from penetrating the membrane surrounding the other species' eggs. Gamete isolation, this is an example. Gamete isolation separates certain closely related species of aquatic animals such as the sea urchins that you see here. These are sea urchins. Sea urchins release their sperm and eggs into the surrounding water where they fuse and form zygotes. It is difficult for gametes of different species such as the red and purple urchins shown here to fuse because proteins on the surfaces of the eggs and sperm bind very poorly to each other. So they physically are unable to have a zygote happen because of this gamete isolation. And that's the last prezygotic barrier. And now we're going to get into the postzygotic barriers. All right, so the postzygotic barriers are barriers that prevent a hybrid zygote from developing into a viable fertile adult. So we have the mating occur, we have the zygote formed, but then the zygote does not develop into a viable fertile adult. So the examples of this, okay, you can see them listed across the top right here. We've got reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. Okay, all of those, like I said, prevent development into a fertile adult. So in the first example, see this little salamander right here? What happens is the genes of the different parent species, they interact in ways that impair the hybrid's ability to develop and survive in its environment. This picture that you see is an example of some salamander subspecies of the genus Insatina that live in the same region and habitat where they may occasionally hybridize. Most of the hybrids do not complete development, and if they do, like in the picture right here, they're very frail. And so they may not live a full life, and then they don't reproduce and pass their traits on to their offspring. The second one is reduced hybrid fertility. I know you guys have seen this before, where even if hybrids are vigorous, they may be sterile. If the chromosomes of the two parent species differ in number or structure, meiosis in the hybrid may fail to produce normal gametes. Since the infertile hybrid cannot produce offspring when they mate with either parent species, genes cannot flow freely between the species. So what you see here, and like I said, you guys have heard this before, is this is a male donkey in the first picture, and then a female horse in the next picture, and then that's a robust but sterile mule below. And so the mule is incapable of producing gametes through the process of meiosis, and therefore is incapable of mating with either the horse or the donkey. And they've even done reverse, where they've taken a hinny, it's not shown in the picture, but it's a female donkey and a male horse, and it is also sterile as well. So that's reduced hybrid fertility. Okay, the last example is hybrid breakdown. It's where some first-generation hybrids are viable and fertile, but then they go to mate with another or either parent species in the second generation or the generation after that, and then they're often feeble or sterile. So strains of cultivated rice, so that's what you're seeing pictured here, have accumulated mutant recessive alleles at different locations in the course of their divergence from a common ancestor. And then hybrids are vigorous at first and fertile, but then the plants of the second generation carry too many of these recessive alleles and become small and sterile, which is what you're seeing in this picture right over here. And so that has, definitely not been a way to have great, awesome, fertile adult plants over time. So it kind of prevents them from having fertile generation after generation. Okay? All right. Okay, Darwin faced many unanswered questions when he began to ponder that mystery of mysteries, also known as speciation. And biologists since Darwin have continued to ask many fundamental questions about speciation. For example, okay, we're looking at this up at the top, how long does it take for a new species to form? And how many genes change when one species splits into two? Answers to these questions are continuing to emerge today. So we can gather information about how long it takes new species to form from broad patterns of looking at the fossil record, also from studying morphological data, which includes fossils, or by looking at molecular data to assess the time interval between speciation events in particular groups of organisms. Okay, the fossil record includes many episodes in which new species appear suddenly in a geological stratum, persist unchanged through several strata, and then disappear. This would be known as what you see here, I'm trying to get my mouse to work right here, called punctuated equilibrium. Okay, so this is one way that some species have evolved today through speciation is by punctuated equilibrium. So it's like a burst of many new species followed by periods of little or no change. Perfect example is there are dozens of species of marine invertebrates that make their debut in the fossil record with novel morphologies, but then change very little for millions of years before becoming extinct. And so in the first little picture you see here, and this is just using a butterfly, but like I said, many invertebrate fossils they've found that do this, you would have this original butterfly that would have a very quick burst of change, but then they would exist like this for millions of years, like you see both of these existing over time for millions of years. That's opposite to the idea of gradualism. Now, we do believe, I say we meaning evolutionary biologists and scientists, believe that there are some things that evolved into new species through both of these methods. One is just very quick with little change for a long period of time. Gradualism is the model that was proposed by Darwin, which is species appear gradually over time. So you can see this first original butterfly that had subtle changes over time and then arose into, these would be current species today. So you're looking at these two species, or up here, these two species could have arisen by either punctuated or gradual patterns. Darwin believed in this gradualism effect. Some guys, in fact, the two men who created this idea of punctuated equilibrium were paleontologists from the American Museum of Natural History. One guy's name was Jay Gould, and one guy's name was Niles Eldridge. And so they proposed this idea of punctuated equilibrium or punctuated pattern. What's interesting, though, I think, is that for species whose fossils changed gradually, as in the method right here at the bottom, it's hard to tell exactly when the new species formed, since information about reproductive isolation does not fossilize. However, it is likely that speciation in such groups occurred relatively slowly, perhaps taking millions of years to have the new species. Okay, now that we have a clearer sense of what constitutes a unique species, let's turn our discussion to the process by which species arise from existing species. Speciation can occur in two main ways, depending on how gene flow is interrupted between populations of the existing species. So the two main ways are allopatric speciation and sympatric speciation. Okay, it's based on, like I said, based on how gene flow is interrupted. So in the first example, allopatric, let's talk for a little bit about this. Allos is from the Greek word other, okay, allos is from other, and then patric refers to homeland, so it's like other home. This is when populations are segregated by a geographical barrier, which can result in adaptive radiation, or what we call island species. So in the first diagram, if you see here, let's say we have this original forest that is separated by the formation of rivers and valleys and ridges, and now what was on this side is now separated from this side. So we have two new populations that exist because they're geographically isolated from each other. This happens a lot when they look at the Grand Canyon. There's certain species of rodents that can't cross the water. I mean, that's the thing, is that if the species is capable of crossing the water, like if it's a coyote or if it's a mountain lions or a bird, then this is less likely to happen with those because they can get across the geographic barrier. But if it's something like a small rodent that can't swim, that doesn't get to the other side, then over a long period of time, the separation of the gene pools causes divergence, and different mutations arise. Natural selection and genetic drift may alter the allele frequency, which leads to this population on this side being very different from this population on the other side. So they're geographically isolated from each other. A lot of times they become so different that now they do not reproduce with each other. That's what happened with the two species of squirrel in the Grand Canyon, is they are no longer capable of interbreeding with each other, and that's based on this geographical barrier. Some other examples, because I like to mention many different ones, but there's the cormorants, which were the island species. When you look at the flightless cormorants, it's thought that they were once part of the flying cormorants, but because they were separated geographically whenever Pangea occurred and the island separated from South America, then you have the species that now, over time, gene flow, genetic drift, mutations, etc., that the flightless cormorant now is very different from the ones that were on the main, South America, and so that's a geographical barrier in a result of adaptive radiation. There's another example in it's called the Andros Island. It's in the Bahamas, where there's a population of a mosquito fish that colonized ponds and became isolated from the other population. So the water levels dropped, and so in this one area, they became separated from each other geographically. And now when they look at these two populations, they differ based on body shape. In one of the ponds, they're preyed upon. There's a predator that attacks them, and when you look at their body shape, they have a more streamlined body shape to be able to swim fast, like quick bursts of energy to get away from the predator. Whereas on the other side, where they're geographically isolated, their body shape is, definitely not streamlined, and it's because it's without predation, and so it just has like a long, skinnier body that's for steady swimming. And so that's allopatric speciation. All right, let's talk about now. Okay, the second way that species can originate or occur is through sympatric. Okay, sympatric speciation. Sym means together in Greek, and so this occurs in populations that live in the same geographic area. So they're reproductively isolated subpopulation in the midst of the parent population. So look at the second example, and my Mobi is getting out on me right at the end of this, so we'll see if my pen works now. Okay, so this example over here, this is sympatric speciation. So you've got your parent population that for some reason has a change in it. Okay, some of the reasons that it could change, it could be gene flow is reduced based on polyploidy, or habitat differentiation, or sexual selection. Okay, so those examples, I'll just do a little bit. I want you to read about these, but I'll just say a little bit. Polyploidy is when a species can originate from an accident during cell division that results in an extra set of chromosomes. And you just think about poly means many, ploidy refers to sets of chromosomes. So plants that are tetraploid, 4N, self-pollinate or mate with other 4N plants, so they isolate reproductively from the 2N plants. So that's been happening for millions of years. It's common among a lot of plant species, so that's polyploidy. Cichlids, another example, are reproductively isolated because of natural selection that results from a switch to a habitat. So it's called habitat differentiation, or it could be a food source that's not used by the parent population, and so then it results in a totally different species. So summing it up, there's two ways of speciation, allopatric and sympatric. And make sure that you review all of this, and I think that's going to conclude our notes on macroevolution.